Hi, my name is Angie Yang, and the genetic disorder I researched on was Tourette's syndrome. Now, Tourette's syndrome is a neurodevelopmental condition characterized by vocal and motor tics. And tics are basically involuntary, sudden, fast, repetitive, non-rhythmic motions and vocalizations. And examples of phonic tics are sniffing, throat clearing, and grunting, and examples of motor tics are eye blinking, shoulder shrugging, and hitting. And in figure 3, you can see the occurrence of tics over courses of time, such as seconds, hours, days, weeks, and months, showing that it is involuntary and they can't control it. And it also occurs with other disorders such as OCD and ADHD, which can be seen in figure 1. It shows a Venn diagram displaying the behavioral spectrum in Tourette's syndrome, which includes OCD, ADHD, depression, impulse control disorders, things like that. Now let's talk about the pathophysiology. PET studies revealed metabolic patterns associated with Tourette's syndrome. One is that there is a reduced resting activity in the striatum and the orbital frontal cortex, and there's also strong activity of the cerebellum, thalamus, putamen, and insula during tic release. Now let's get into the genetics portion of Tourette's syndrome. And Tourette's syndrome is caused by the bilineal, which means that it's from both the maternal and paternal sides of the family. Transmission of susceptibility loci. And only a few genes have been reported, and I will talk about that more. But the efforts to definitively identify Tourette's syndrome's genetic foundation are hindered by its complexity. Now let's talk about the underlying gene locus. Only a few genetic loci have been identified. One is the SLITKR1 gene, and it codes for neuronal transmembrane molecules. And the mutation leads to unusually shortened, non-functional forms of protein, and a single base pair in the section of DNA close to the gene is altered. The second genetic loci is L-histidine decarboxylase (HDC) gene, which codes for the rate-limiting enzyme in histamine biosynthesis. And basal ganglia abnormalities can be seen in HDC deficient mice, which was in a study in one of my articles, which include the histaminergic regulation of dopaminergic modulation and information processing. And in figure 2, you can see the potential mechanism of activities, the abnormal activities in HDC mice. Now let's discuss the treatments and future direction. One treatment discussed in my article was habit reversal training and exposure response prevention. And another treatment that was frequently discussed in all of my articles were anti-tic drugs. One was haloperidol and pimozide, which are suppressants, but they do have side effects. Another drug was typical antipsychotics, alpha-2 adrenergic agonists, which include clonidine and guanfacine or facine, I don't know, and botulinum toxin injections. And the role of the basal ganglia, which was previously discussed, which is involved in motor control and in the mechanism of Tourette syndrome, should be further explored, and also their connections to the frontal cortex and their brain structures should also be further explored. And in figure 4, it shows the therapeutic strategies for Tourette syndrome. You can see that has tics, OCD, ADHD, which were also mentioned as other genetic disorders that Tourette syndrome, people who have Tourette syndrome also have. And you can see the different lines of strategies. That concludes my Tourette syndrome research. Thank you.